Hi, everyone. Welcome. We're going to give everyone a moment to join and then we'll get started. So just bear with us for another minute and then we'll get going. If you're just now tuning in, as I mentioned, we're going to give everyone a minute to join the webinar and then we'll get started. So probably just another 30 seconds or so, and then we'll get going. All right, I know we're likely going to be over capacity today, so I think we can go ahead and get started. If you have any friends or colleagues who are not able to join because we reach capacity today, as we said in our emails, please feel free to communicate to them that everyone will be able to watch the webinar after in its entirety. We'll send a link out so everyone will have access. If this is your first time tuning in, we're very excited to have you here. You're in for a real treat today. And if you've joined us before, we're glad you're back. For those of you who don't know me, I'm Whitney Christopher, and on behalf of Elysium Health, I am thrilled to welcome you to today's webinar, Unlocking Longevity, the Science of NAD and Sirtuins, led by Elysium co-founder and Novartis professor of biology at MIT, Dr. Leonard Guarente. Among his family, friends, and colleagues here at Elysium, he goes by Lenny, so I'll refer to him as Lenny today. As I shared at the top of our last webinar, one of the best parts of my role here at Elysium is the time I get to spend working with our amazing science team and renowned scientific advisory board, and that includes my friend Lenny here. If you're newer to NAD and sirtuins, which are often referred to by scientists as our longevity genes, then you're definitely in the right place. Maybe you've seen NAD and sirtuins on Instagram or TikTok or TV. It is everywhere. And you are in the right place, as I said, for today's event, where you can trust Lenny to help you separate fact from fiction. If you're a longtime Elysium customer or you've been following Lenny's research at MIT for all or part of the last 4 decades, then I don't need to tell you that he is an absolute legend in aging research. Since he won't sing his own praises, I will share with you that around Elysium, he is lovingly referred to as the Godfather. Not that kind, of NAD and sirtuins. So don't worry about that. Many of the biggest names in longevity, apart from his own, have come out of Lenny's lab at MIT. Before I turn it over to Lenny to start his presentation, one small bit of administrative housekeeping, as is our usual format. If you have questions that you want to ask Lenny that he will respond to personally, please just enter them into the chat function and we will film that and follow-up content at a later date. We'll send that out via our newsletter and share it across Elysium's social media channels. So if you forget to submit something today during the webinar, you think of a question later and you can't submit it in the chat function, just send us an email. It'll be on the slide at the end of the presentation to events@elysiumhealth.com, and we will make sure to get that question answered for you. So without further ado, please join me in welcoming Lenny as he endeavors to help us unlock our longevity with the science of NAD and sirtuins. Lenny, take it away.
Thank you, Whitney, and hello out there. So today we're going to focus on what I think is a very promising intervention to slow down the aging process, which is NAD and sirtuins. Now I'm going to begin with a more general introduction into what is aging. So depicted here is a survival curve as it would have looked 150 to 500 years ago, where the average human lifespan was maybe 30 to 40 years old. And it looks basically like a decay of a radioisotope. So the sort of random or stochastic death of people over time. And the reason for this is that people died for all kinds of reasons. For example, being accosted by robbers in Sherwood Forest, or by being hit by an infectious disease. And these diseases were rampant at that time. So people didn't live very long and they died randomly. Now, if we skip ahead to the present age, we see the average lifespan is around 80 plus or minus a few years. And we see the curve looks like it has a different shape. It's been rectangularized. And that's because death nowadays is mostly due to aging. And the reason is because of the successes in sanitation and sterile techniques, vaccines, medicine, job safety, childbirth, etc., to prevent random deaths. So that most of us now survive until aging takes its toll. And the reason there's this sharp drop-off in survival here is that mortality rates are going up as we age, so that by the time we reach this cliff, as it were, mortality rates are very high. They have been increasing exponentially, and aging on a population level becomes precipitous. Now, aging is important, and the reason it causes all these deaths is that it's the biggest risk factor for the major diseases: cancer, Alzheimer's, heart disease, diabetes, dementia, and others. So if we could slow down aging even a little bit, it would have a major impact on all of these diseases and would really be transformative for society as a whole. Here's an anecdote I want to share with you about a woman who's the longest lived person ever to be documented as such, and her name is Jean Louise Calment, who lived to be 122 years old. This is a picture of her on her last birthday. And the story I want to share is this one. In 1965, when she was 90 with no heirs, she signed a deal to sell her apartment to a lawyer on a contingency contract. And the contract said that the lawyer, then age 47, agreed to pay her a monthly stipend of 2,500 francs until she died, when he would move in. And the lawyer ended up paying Calment the equivalent of more than $180,000, which was more than double the apartment's value. And after his death from cancer in 1995, his widow continued the payments until her death the following year. So why can't we all live to be that age? And that's because aging is a complicated process. Here's a review called The Hallmarks of Aging. And hallmarks are really causes of aging. That's how I like to think about it. That was published in Cell. And the major causes that these authors identified are shown here: genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, disabled autophagy, dysregulated nutrient signaling, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, altered cell communication, chronic inflammation, and dysbiosis, which boils down to how the microbiome is treating you. Now, we wrote a review published this year, not on causes of aging, but to review the status of interventions that could slow down aging. And we reviewed human trials that have been published or that were in progress and identified basically the 8 most promising interventions based on the literature and what was actually taking place in the clinic. And they're shown here. One is metformin, which was known for some time as a treatment for diabetes and which reduces ATP production in mitochondria. Another is NAD sirtuins, the topic for today, which has functions both in the nucleus and in the mitochondria. GLP-1, which is an important protein in metabolism and which controls, among other things, satiety. And these GLP-1 receptor agonists, for example, Ozempic or Wegovy, have the effect of actually causing weight loss in people who are overweight and exerting numerous metabolic effects that result. And it's very likely this will impact aging in these people. Another is rapamycin, which inhibits TOR, and this reduces protein synthesis to kind of slow things down. Another is spermidine, which is an inducer of autophagy, which clears up damaged protein, and mitophagy, which clears damaged mitochondria. Another is senolytics. These are compounds that will selectively lyse senescent cells without being harmful to normal cells. Another is probiotics. And the idea here is to get rid of these bad species of bacteria that colonize the gut and that cause bad health consequences, such as inflammation. And the other is inflammation writ large, anti-inflammatories, to reduce the effects of these very active products of the immune system, which fight infections, but which also cause tissue damage, interleukins, TNF-alpha, and so on. And today, the focus is here on NAD sirtuins. So I thought I would begin with an introduction, historical introduction of how we got involved in this work. And this is taking us back to the early 1990s and 2 graduate students, Brian Kennedy and Nick Austriaco. And we started by studying aging, replicative aging in budding yeast, which is what my lab worked on at the time. And in this system, cells divide so that you get a mother cell, which consists of old material, and the daughter cell, which consists of newly synthesized material. And what you can do, and this was established already as a system, we didn't invent this, was follow the same mother cell through multiple divisions in the microscope on a Petri plate. And what you found is that the mother cell goes through roughly 20 generations and then stops dividing. And the cell has reached senescence and also looks large and old at that point. So this was a kind of an aging process. We chose it because we thought it was accessible to the kind of science we wanted to do. And I'll just point out that the 2 students, Brian Kennedy and Nick Austriaco, after they left my lab, took rather divergent paths in life. So we did a genetic screen to identify genes that could make these mother cells live longer. And the key gene that emerged from the study is SIR2. And here's an example of why this gene controls aging. A normal strain had 1 copy, and this is a survival curve of mother cells now. You can see it follows a normal aging curve here with about 20 generations determining half survival, 50% of the cells are dead. If you get rid of the SIR2 gene, knock it out, you shorten the lifespan. And if you had an extra copy, 2 copies now, there's a long lifespan. So this convinced us that at least in yeast, this was an important gene in aging. We wanted to know what SIR2 did. And we also knew at that point that there were homologs of SIR2 in virtually all species writ large, and that includes humans. And so we're interested biochemically in studying the SIR2 protein. And we were studying the yeast and the human protein in these experiments. And these were done by Shin Imai, who was a postdoc in the lab at the time, is now a professor at Wash U. And he and I went through this year after year, and finally we hit upon something. So it was thought that maybe SIR2 proteins had something to do with deacetylation of histones, and that's what we were kind of testing. And nobody, and other people were studying this, and nobody could demonstrate any activity in vitro. And the reason was there was a missing ingredient, and that missing ingredient was this guy here, NAD+. And if you have NAD in the reaction, you see this product peak of a deacetylated peptide, okay? I don't show you, but if you leave the NAD out of the reaction, nothing happens. So these proteins then are NAD dependent deacetylases. And we made that discovery in 1999, I think an important discovery in the field. And that means that the proteins bound to DNA, histones, are acetylated largely on their amino termini. And SIR2 and SIRT1, it's homolog in humans, it's closest homolog, deacetylate this DNA protein complex called chromatin, and the NAD actually gets split in the process, and the deacetylated chromatin wraps up in a tighter form and is silenced to gene expression. Now, we thought that this was a pretty interesting activity, and we speculated that it might explain or relate to something that had been known for, well, now it's about a century, called calorie restriction, in which rodents, given a calorie restricted diet, live longer. So why do these mice or rats live longer? There's an example in primates to show that this is very likely true in all mammalian species. So this is primate number 1 that was fed ad libitum, and at the same age, primate number 2 that was calorie restricted. And there's a pretty big difference on in their appearance, in their activity level, and in how long they're going to live. The calorie restricted primates live longer. So we think this is a conserved pathway. And we're interested in posing the question, does calorie restriction, in order to work, create longevity, does it require SIR2? So this is the experiment we did in yeast. These curves here are shown on high glucose, which is kind of the yeasty way of calorie restricting. And if you lower the glucose 4-fold, the mother cells live longer, okay? Sorry, that's here. This is the normal cells with high glucose and low glucose. If you knock out SIR2 over here and you do the same experiment, cells can survive on the lower glucose, but they don't live longer. You don't increase the lifespan. Okay, so we believe this works in yeast. We could also, using a genetic trick I don't have time to describe, ask whether silencing by SIR2 increased. And if this is a wild type strain on high glucose versus low glucose. And the redder color of the colony is indicative in this strain of increased silencing by SIR2. If SIR2 is deleted, you don't see any effect of the low glucose. So this was the first hint then that calorie restriction quite generally might require SIR2. And I should say that now in mice, there's a lot of data to support this idea that knocking out a SIR2 homolog in mice will abolish one or another aspect of calorie restriction on the effect on the physiology of the animal. Okay, now I want to describe what's going on in humans or in mammals in general. There, in mammals are 7 SIR2 homologs shown here. The orange are nuclear proteins, SIRT1, 6, and 7, 3 of them. The blue are mitochondrial proteins, SIRT3, 4, and 5, and these are mapped onto a phylogenetic tree of other species, but that doesn't really matter. And then T2 is cytoplasmic. So the SIRTs are the 7 human homologs of SIR2. And the field has blossomed considerably since the early days of this research, and is now, and has been for some time, quite widely studied system. So Sirtuins can extend lifespan in yeast. I showed you in C. elegans and flies. There's some controversy. Early studies showed in both C. elegans and flies that increasing the dosage of the relevant SIRT2 gene extended the lifespan. Now, those studies were challenged about 13 years ago in a paper that said that wasn't true. And since then, there have been many studies that validated the original studies that show that SIRT2 homologs in the roundworm, C. elegans, and in flies do indeed extend the lifespan when their activity is increased. So there's yeast, there's C. elegans, flies, and mice also. The SIRT6 gene, one of the 7 mammalian homologs, was shown by Cohen in his lab to extend lifespan in mice when overexpressed. And in humans, intriguingly, in the past year, it's been shown that there is a polymorphism in the SIRT6 gene that changes 2 amino acids in the protein that is highly enriched in centenarians, which are people who live to be 100 years old. So that's a very exciting finding that really is suggestive that the longevity function of the sirtuins, which is SIRT2 and all its homologs, might have been conserved across all organisms, including people. Okay, so how might sirtuins slow aging? Okay, well, epigenetic silencing of the genome by SIRT1 and SIRT6. If you deacetylate the histones, okay, you exert an epigenetic effect that results in silencing of the genome, and that could well explain part or all of the effect on longevity. Repair of DNA damage, I didn't talk about it, but SIRT1 and 6 have also been linked to DNA damage repair as positive regulators of repair, and a lot of that work in Gorbanova's lab, and that may well also contribute to longevity function. Preservation of stem cells, SIRT1 has been shown to slow down the death of stem cells in aging animals. Another, and that all those are nuclear in their effects. In mitochondria, SIRT3 is involved in deacetylating proteins involved in metabolism, and also in detoxifying reactive oxygen species. So scavenging oxygen radicals and effects on metabolism, which is exemplified by fatty acid catabolism, may also be important in promoting longevity. Another possibility, number 5, is activation of autophagy and mitophagy, which I mentioned earlier by SIRT1. And finally, SIRT3 and 5 in mitochondria can remove larger acyl groups from proteins, which get there because there's a high concentration of acylated CoA intermediates in the mitochondria, and that might be important. So these are kind of more molecular models. How would these map onto the causes of aging, the hallmarks that we talked about earlier? So does sirtuin activity affect the hallmarks of aging? And indeed, among these thousands of studies on sirtuins, there is evidence that there are sirtuins that affect genomic stability, SIRT1, 3, and 6, telomere attrition, 6, epigenetics, proteostasis, autophagy, nutrient signaling, mitochondrial function, cellular senescence, and so on and so forth. So I think there's a good concordance of sirtuins being effective in targeting these hallmarks of aging. And they do this by by molecular activities, most prominently deacetylation or deacylation of proteins. Okay, what about diseases? So it's not that easy to know in humans unless there's a genetic variant that causes a disease, whether a gene is linked to a disease, but one can ask that question in mouse models. for various diseases. And that's been looked at for sirtuins, and they've been shown to affect cancer. Okay, so loss of function mutations in sirtuins promote cancer, diabetes, neurodegenerative diseases, Alzheimer's, Huntington's, and Parkinson's disease, cardiovascular disease, and one of the prominent effects here is SIRT1 has a protective effect on endothelial cells that line blood vessels, osteoporosis, kidney disease, liver disease, and inflammation. So, at least from the rodent models, there are lots of activities to look at in people and diseases to look at in people with respect to the protective effects of sirtuins. And I'll get to some of that later in the webinar. So the question then is, are sirtuins actionable? And I want to start with really the first finding that show that sirtuins can be activated exogenously. So in humans, it's still not really realistic to do gene intervention to change the activity of a gene. So one looks for small molecules that can change the activity of a protein. And the first indication that such existed for sirtuins came from work by David Sinclair, who showed that a molecule called resveratrol in red wine had a protective effect on activity of SIRT1. So it could increase the activity in the test tube, and it could improve the health of mice, especially when stressed with a high-fat diet. And that effect on the enzyme by resveratrol is very likely due to direct binding to SIRT1 and not by binding to SIRT2 through 7. So resveratrol 15, 20 years ago was a very promising molecule for humans. And there have been many trials now that have been done, and it doesn't work so well in people. The trials are very spotty in their results. And what seems to have emerged from the literature is a related compound called terastilbene, or PT, which differs from resveratrol only in that two hydroxyls have been replaced by a methyl group. And this also is a natural compound, which is found in blueberries and not wine. So that sort of said that sirtuins could be activated, and we think this is still a promising avenue for activating sirtuins to slow down aging and improve health. But what's really overtaken this field is NAD itself. And I think a key finding here was the finding that NAD is depleted with aging across model organisms. This was first shown in a paper from Jim Imai's lab in mice in 2008. Now here's a later study we collaborated on in 2013 with Johan Allwart's lab. And what they show here is that clearly, as predicted, NAD levels go down in aging mice. So this is liver here, young versus old, and this is muscle, young versus old. So NAD goes down about twofold. Is that enough to affect the activity of sirtuins? So we could look at the activity of SIRT1 in particular, because we know it deacetylates a protein called PGC1 alpha, which happens to be involved in mitochondrial regulation. And we could look at the acetylation status of this protein by probing a blot with antibody to acylysine. And you can see here that the acetylation is higher in old mice, which means the deacetylation by SIRT1 is diminished in old mice compared to young mice. So indeed, low NAD is associated with low SIRT1 activity. And of course, that's bad news as far as slowing down aging. Now, why does NAD go down? We still don't know for sure. One possibility is that it's chronic activation of proteins like PARPs that deplete NAD. DNA damage will trigger PARPs to do that, or upregulation of another NAD degrading enzyme, CD38, or NAD synthesis is linked to the circadian clock. So if the clock gets less active, with aging, that also would reduce the synthesis of NAD. So while we don't know yet why this happens, we know that it does happen. So the idea is then, if NAD is going down in aging animals, including humans, can you replenish it? And the possible candidates to replenish it are NAD itself, okay, or precursors of NAD. So here's the NAD molecule. This is nicotinamide riboside here, NR, and with the phosphate attached, that's nicotinamide mononucleotide, or NMN. And indeed, it was shown by Charles Brenner that NR could drive NAD synthesis in cells. So it was a possible precursor of NAD that might be able to increase NAD in vivo. And Shin Imai and his associates did the same for NMN and showed that it could also drive NAD synthesis via this pathway shown here. And so exogenous NR or NMN could increase NAD in cells or in the animal, and that could upregulate the activity of the sirtuins and slow down aging. So that's the kind of the promise of this approach of the NAD pathway and sirtuins to slow down aging. And what I want to say, though, point out and stress, NAD itself does not get into cells, whether orally applied or by IV, but NR and NMN do. So in the same situation where NR and NMN will drive increases in NAD in vivo, exogenously applied NAD, whether it's orally or IV, will not. So I would stay away from NAD supplements or NAD IV infusions because they're not likely to work. Now I'm going to turn to more recent findings, some involving Elysium Health. And I want to pose the question, well, now I've mentioned two different ways to activate sirtuins, NAD boosting, say by NR, and direct activation of a SIRT1, say by resveratrol or terastilbene, okay? Now, what if we did both? What would happen? And we hypothesized years ago that the combination of these two would work synergistically. And there are two studies now that suggest that this is indeed the case. One is a study by Rick Grantstein's group at Cornell. And what they did is looked at the effects of UV irradiation on a patch of skin in a mouse, a live animal. And they showed that the radiation at a given dose would induce measurable outputs, including swelling at the site of radiation and immune suppression. And what they found is that nicotinamide riboside or terastilbene by itself would partially ameliorate the swelling and the immune suppression, the effects of the radiation dose. But the combination of the two, NR and terastilbene, had a much bigger effect, much bigger protective effect. So that was a demonstration of synergy in an in vivo model. In a different paper, it was shown by Jose Estrella and his colleagues in a mouse model for ALS, the SOD1 mutant mice, that NR and terastilbene cooperatively delayed motor neuron dysfunction in that mouse. So both of these studies are consistent with the hypothesis that adding two boosters that work by two different mechanisms on the sirtuins can have a protective effect that's bigger than the effect of one or the other. Now we at Elysium were really interested in this point because the first product of Elysium was Basis about 10 years ago, not quite, which combined NR and terastilbene. So we're really interested in knowing whether this pathway is a good target in humans. And we now know that it is, but years ago, we needed to begin some of the legwork to get at this question. And the first human trial that was done on basis by Elysium was a repeat dose trial that showed, so here's the thing. You want to add these NAD precursors, and what you want to do is boost NAD in vivo and show some beneficial effect on health or aging. You first have to show that you've really boosted NAD by these molecules. That's a key milestone. And what the study was aimed at then is to test this hypothesis. So it was a human trial, randomized, double-blind, placebo-controlled, okay, as all our trials and all good trials are, involving 120 people, duration of 8 weeks. Primary endpoint was, again, to get still more evidence regarding safety. And the secondary endpoint was, do we really boost NAD levels in humans? And the results were extremely clear-cut. So it's a 2-month trial. We took data points at 1 month and 2 months, and you could see that NAD is clearly boosted. This is the recommended dose of basis shown here, and this is doubling the dose shown there at 1 month, and at 2 months, it kind of you reach a steady state where the two doses are pretty similar in the boosting that they give. And what's really interesting is that the degree of this boost is roughly equivalent to the NAD that's lost in normal aging. So this is really showing, the people in the study were 60 to 70 years old. So this is really showing that NAD boosting by basis is offsetting and not overshooting, at least not greatly overshooting, the amount of NAD that's lost over normal aging. And we're very excited by this study. So we wanted to look at a study that would get more to the question of disease. And we're interested in disease of the liver because in rodent models, sirtuins had shown a protective effect in the liver. So the model we looked at is shown here, which is non-alcoholic fatty liver disease. And what happens in this model is that the normal function of the liver, which is to make lipids, among other things, becomes dysfunctional, and the liver gets inflamed, and that's goes from NAFL. If the inflammation gets strong enough, it goes to NASH, which is steatosis of the liver, and finally to cirrhosis, which is a diseased organ with a lot of fibrosis. And this NAFLD, then, this stage here, is characterized by a fat buildup in the liver associated with obesity. So this is really common in obese people. It's thought that something like 100 million Americans might have this fatty liver disease. And this is the progression here. And as we're progressing, what's happening is obesity or metabolic syndrome is triggering mitochondrial dysfunction, oxidative stress, and eventually apoptosis, where hepatocytes, liver cells, are actually lysing in vivo. And that's causing and being caused by pro-inflammatory cytokines, which are attracted to the liver by the fat deposits. And you get this vicious circle of inflammation and cell death in the liver, leading to cirrhosis. Okay, so the study then is, which was published last year in Hepatology, was 111 subjects, again, randomized, et cetera, et cetera. And we looked at a duration of 6 months, and the two endpoints were a primary endpoint, which was liver fat content, how much fat was in the liver. We only enrolled people who had fatty liver. And the secondary endpoint was inflammation, which we could measure by various blood markers. And the result in the trial, which was quite interesting and unexpected, is that the basis group really had no significant change in fat content in the liver, which on the one hand was disappointing. On the other hand, was offset by the fact that even though the fat wasn't reduced, the inflammation, the effect of the fat, was reduced. Inflammation was reduced in these people, which is very exciting to see. So BASIS reduces markers of hepatic inflammation in non-alcoholic fatty liver disease. So what is, so again, this just reiterates primary endpoint, fat content, not a significant change. Secondary endpoint, liver inflammation showed an amelioration. Now, how do we know that there was inflammation, and how do we know it was in the liver? Well, really the gold standard here is something that's measured in any good physical that you'll undergo by a physician, which is the presence of a liver enzyme in the blood. So this is an enzyme called ALT, which you can see here. And we looked and compared in the placebo group versus the NR terastillabine or BASIS group. Was there a difference over 60-day dosing in ALT in the blood? And the answer is yes. There was a much bigger reduction in ALT in the treatment arm than in the placebo arm. And the significance was quite high regarding that difference. And we're convinced of its significance because the same was found for two other enzymes in the liver, AST and GT. And what that means is that the treatment arm were experiencing less liver damage and lysis of liver cells, which would release these enzymes into the blood for detection. We did a second measurement of something quite different, which is a toxic lipid species called ceramide, shown here. And we found that a particular ceramide species called 14.0 was significantly reduced here in the 6-month trial in the treatment arm, but not in the placebo group. And that was very consistent. And you could, won't show the data, but you could go patient by patient here. And what you find is that the people who showed the reduction, the biggest reduction in ALT, the liver enzyme, were the people who showed the biggest reduction in this toxic lipid species. So this is a result I think is very exciting from the NAFLD trial. BASIS lowers multiple markers of inflammation in these NAFLD patients. And that means BASIS may prove useful to treat NAFLD patients and slow progression to NASH. And if you can slow that progression down that pathway of doom, you're protecting these people against liver disease. Finally, BASIS, so in these people with obesity and metabolic syndrome, fat is accumulating not just in the liver, which is what we looked at here, but in other places too. And it's possible that BASIS may favorably impact other organs in people with metabolic syndrome. Okay. I'm going to turn to a more recent trial that was, I think, published just last week on a disease called COPD, which is chronic obstructive pulmonary disease. This is an ongoing lung condition that's caused by damage to the lungs. The damage results in swelling and irritation and inflammation inside the airways, which limits airflow into and out of the lungs. And emphysema and chronic bronchitis are the two most common types of COPD. So the study that was done by a group in Denmark, and we collaborated with that group on this study. And the trial then had 40 COPD patients and 18 healthy controls. And this was an NR only trial, so an NAD boosting only trial. It doesn't have the terastillabine SIRT1 activator. And the arms were, of course, placebo in the NR group. And it was a 6-week trial with a follow-up period. Now, the primary endpoint here was interleukin A, or IL-8, in saliva. And the reason for that is IL-8 is the main pro-inflammatory cytokine, which is upregulated in COPD and which is linked to the pathology, the lung pathology of COPD. And here are the results of the trial. So again, it's NR versus placebo, very simple data. And at the end of the trial, here there was a reduction, large reduction in interleukin 8 in the treatment group, the NR group, and not in the placebo group. So the primary endpoint of this trial was met. NR significantly decreased IL-8 in the sputum at the end of the trial. Other data I don't have time to go through, but I'll mention also significantly decreased the number of neutrophils in the sputum, which is related to IL-8. IL-8 attracts neutrophils, and this all contributes to the pulmonary damage in these people. So the conclusions of this trial is that IL-8 was significantly reduced by NR. NR reduced airway inflammation. I don't show you data for this, but one can look at other markers of inflammation. So what does this mean for healthy people? NR may support lung function more broadly. This will be a very interesting subject to look at going forward regarding other diseases, for example, emphysema, and so on and so forth. Okay. So those are a flavor of specific trials that have been done. Now, in collaboration with other institutes, Elysium's demonstrated that NR bases may confer health benefits in human trials on Parkinson's disease, ALS, to neurodegenerative diseases. Parkinson's is very interesting and has passed through phase 1 now, which gave this hint of benefit up to a phase 2 trial, and phase 3 is planned upon that, because there are a lot of people with Parkinson's disease who really are in need of a treatment better than the state of the art, which is dopamine. ALS, which is a devastating disease in which people have a fairly short life expectancy after diagnosis of several years. And a pilot study done there looked promising. And the study was done some number of years ago, I think 6 or 7. And I believe, and it was a small trial, but I believe 6 of the people who were taking, it was a high dose of basis in that trial, are still alive today, which is small, so it still has to be viewed as anecdotal, but I think highly promising, and a larger scale trial is in the works. AKI, which is acute kidney injury, turns out that something like a third of patients who go through major surgery, major cardiac surgery, have kidney failure after the surgery. And in mouse models of this, sirtuins have been shown to be protective, SIRT1 in particular. So this is, again, a major unmet need. And we've done a phase 1 trial in collaboration with Mass General Hospital on this, and also with Mayo Clinic. And it's certainly safe to give patients basis after surgery. And there's some hints that it may be helping, but I think a larger trial is badly needed for AKI. There's a pilot study on menopause that suggests that the symptoms might be ameliorated by basis. And again, what we're gearing up for here is a larger trial, more extensive trial to back this up. So in general, of course, more work needs to be done to confirm and extend these findings. But I think there's quite the prospect that there'll be a number of indications that can be treated in this way. And lastly, I'll just mention here that Elysium has also shown in an observational study that people who take basis for 2 years are younger biologically. And the way that's younger biologically is kind of a charged term. So by your biological age is really how healthy you are. So the way I put it is your chronological age is how long you've lived, and your biological age is how long you're expected to live given your age. Now, test that is done is a DNA methylation test. And there's really been a revolution, I think, in biomarkers of aging in the past 10 years due to DNA methylation assessment. This is pioneered by Steve Horvath. And what's done here is to measure the methylation status across 1 million sites in the genome. And that methylation varies with time. And by measuring it, you apply machine learning and an algorithm to set methylation to the biological age. So you get this tool that can then measure biological age. And there are numerous clocks of this type out there. One of them is called Index, which is the clock Elysium developed, and that was used here. And this clock has performed very, very well up against the other clocks. And we published a paper in Aging Cell recently that compares these clocks. But this data I'm referring to here was with Index. And the finding, again, is that people who take basis look like they're biologically younger than people who don't take basis, even though they're age matched. Okay. So in summary, then aging is the biggest risk factor for all major diseases. This is a summary of what I just told you. 2, there are 8 aging interventions are currently being explored in human trials. 3, sirtuins and NAD, which is one of the 8, NAD sirtuins and NAD, are promising longevity targets, and I told you why I think that is so. The 4th is NAD can be restored, so this is a really actionable pathway. Can be restored to youthful levels in older adults. And 5, we're starting to get some of the returns now, early data on human trials. And NR and NRPT or basis ameliorate inflammation and non-alcoholic fatty liver disease and also COPD, and may actually be useful in treating these conditions and perhaps other conditions. So in the last minute or two, let me just broadly state what I think the future of aging research might be. So we focus today on NAD sirtuins, but there are also the 7 other interventions we discussed. And I think these will be thoroughly vetted and tested in human trials. So we're going to know which one of these work well, which ones don't work, which ones might actually be bad for you. And I think that some good is going to come of this for sure. And I think, as I said earlier, NAD sirtuins stands high on the list as far as promise is concerned. Now, what about things other than compounds, supplements, or drugs? What other approaches in the aging field might turn out to be profitable? And I think one thing that's been underutilized so far is big data. I think there's going to be a huge databases that follow very large cohorts of people, numbering in the millions. And I think that the data can potentially be leveraged for preventive, personalized health that can extend health span and perhaps lifespan as well. And I think that mining big data, we really haven't gotten much out of it so far. But I think it's because there's a lot to be had there, but it's going to take time, and it's going to take really large cohorts to tease out what information we can regarding how people age. The second area I think is promising is cellular reprogramming. And I think this is Yamanaka's breakthrough, where he showed that adult somatic cells could be reprogrammed back to cells like embryonic stem cells that he called iPS cells by simply adding to those cells 4 genes. And those genes could convert cells backwards in time. And there's some indication now from Belmonte's lab that something like this can be done in mice, where transgenes for these 4 genes can be induced at the right level and at the right time to reverse or at least slow aging in these animals. So I think this is highly promising. And, the word reverse aging, it's been kind of an oxymoron in the past because we always talk about slowing it. Nobody ever thought about reversing it really, except people who write movie scripts. So I think that it will be really fascinating to see how this plays out. But realistically, this appears decades away from safe application. But I think a little bit of luck, it might be possible to do this. And the trick would be to do it somehow, figure out a way to do it with compounds or drugs if possible, rather than genes. But I think in particular cases, say for particular niche tissues, which can be accessed for gene intervention, it might be possible to have beneficial effects on aspects of human health. So I'm going to close now and thank you all for being here. And we're not going to do a Q&A period now, but please submit questions to this email address, events@elysiumhealth.com. And I think Whitney is going to say a few words here, perhaps.
Thank you so much, Lenny. That was fascinating. You shared a lot of information, and we have received an amazing amount of questions from the attendees today. So as I mentioned at the top of the talk, we will be compiling all of those questions, reviewing them with Lenny. He will answer them personally, and we will share that follow-up Q&A via our newsletter and our social media channels. So feel free to continue to drop your questions into the chat here or to submit them via email to Elysium events@elysiumhealth.com. And thank you so much for attending today. We look forward to seeing you at our next webinar. And thank you again, Lenny. This was fantastic.
Thank you.